Ladies and gentlemen, welcome to Genomics Talk, where we discuss the latest developments and cutting-edge innovations taking place within the genomics community. With us today, our panel of scientists include Damien Arena, Mia Liu, Conrad Leon, and Sarah Eckenrode. And now, here's your host of Genomics Talk, Amanda Perel.
Question, Meow. I'll direct this to you. With the advent or arrival of synthetic biology, do you foresee synthetic therapeutic antibodies in igniting the next wave of therapeutics?
Yeah, let me start. So, for the therapeutic antibody, I think it's one of the fast-growing field in the biopharmaceutical industry. And historically, hybridoma and phage display technology has been very popular in this field. As everybody knows, hybridoma technology has been in this field for a long time and has been very popular, but it does have its own drawbacks. And the new synthetic library approach combined with the phage display technology has been starting to get popular in the past 10-20 years. For the synthetic library, the advantage of that to make engineered antibody is it not only limited to the antigen that we used in the initial immune response in the animal models. For this synthetic library approach, we can artificially design the library and do the nucleotide randomization based on our design or knowledge about that antibody. Then, as everybody knows, for the synthetic antibody approach, there's two things very important. One is the accuracy of the library, one is the diversity of the library. And for the accuracy of the library, that's primarily based on the how we do the oligo design and the oligo quality and how we assemble the oligo into the library. And for the diversity of the library is also primarily based on how we do the PCR assembly of the library, and also the limited to the efficiency of the host content cell we use for the transformation. And the last and the most important I want to mention for this new synthetic library approach to make antibody is the cost. For the past 10 years, the cost for the gene synthesis has been dropped a lot. And this is give the researchers in this field a very good advantage to synthesize more genes and more synthetic libraries. Especially right now, I do see a researcher synthesize like thousands of genes individually, those antibody sequences for their downstream screening. This is a more targeted approach to study the antibody functionality, and this will give the customer, I think, will speed up the whole process for the researcher to find a good antibody for their study.
Okay, great. Thank you. Good answer. I think the next one, Conrad, may be a little bit more geared for you. PCR bias tends to play a role during screening. What efforts are being done to address this type of issue?
I think PCR bias is not really limited to screening. Basically, there's so many molecular biology works that are using PCR. And unfortunately, there's no simple answer to that, right? So basically, if you look at the PCR process, right, everybody, I guess if you're familiar with the PCR reaction, so the less cycles you run it, usually less bias would that be. But at the same time, I think selection of enzymes is also very important because some enzymes definitely have higher processivities, meaning that they won't fall off as much, so that you will reduce certain type of bias. But at the same time, I think for antibody engineering, you want to look beyond just PCR because at a certain step, you want to start with reverse transcriptions. And then actually reverse transcriptase also have bias too. Many people have not really looked into it, but we did some research on it. Based on different selection of reverse transcription, you end up having different diversity of the libraries. So I think people should be cautious about it to look into, see what kind of enzymes you want to do for PCR, what kind of reverse transcriptase you want to use. So definitely this can reduce your time in terms of like wasting time in producing libraries that is not diverse enough. And also we learned it also from very, I would say, unfortunate experience is that actually for PCR bias, one thing we learned a lot about in enzyme engineering that happens is strand switching. Not many people mention about it in the research field because people just look at errors and then whether you have certain type of GC which get amplified less frequent. But actually, because when you do antibody library preparation, a lot of the time you have very similar sequence. And then when you get very similar sequence, when you do a PCR reaction, you will start to switch from one strand to the other strand. And then that's pretty crucial for antibody work because you want to have reliable heavy chain and light chain pairings. But if you're switching strand, that means that your pairing would not be as what you are looking for. So to overcome that problems that we have been working on different type of enzyme, again, like what I mentioned before, enzyme processivity is very important. So we find that actually certain enzyme with high processivity definitely can reduce it. And also to get a reduced number of cycles, we can reduce the strand switching in terms of like frequency down to like single-digit levels. So I think before you start the experiment, I guess definitely look into what enzyme you want to use, from reverse transcriptase, from PCR polymerase, and those definitely two things, and reduce the number of cycles and time too.
Okay, great. Some good advice there. All right, the next one I think may be ideal for Damien to respond here. Standard NGS sequencing technologies have size limitations that impact full-length antibody profiling. What is being done to address the sequencing gap here?
Great question, and very exciting news on that front. So typically, short read technology is commonly used to target very specific regions within antibody libraries, for example, variable regions for the light chain or the heavy chain of an antibody. For customers that are looking to pair the variable regions of that light chain versus the heavy chain, there has been a big pain point through next generation sequencing technology, which is the size limitation. Recent updates from PacBio, who's one of the leading technologies out there for long sequencing through next generation sequencing technology, is that they're able to sequence contiguously through multiple kilobases of DNA. So this is now opening up the doors for antibody discovery research to allow scientists that are doing in vitro assays, like phage display, to actually sequence through the whole region. So they can start looking at the variable region on a light chain and pair that with the same variable region of that antibody on the heavy chain. This hopefully will open the doors for the antibody discovery community to actually expedite discovery of these new targets that they're looking for.
And I think this is a good example to show strand switching problems, because when you amplify, let's say 2,3 kb with a vector backbone that's so similar, it tends to switch too. So definitely when you do NGS work, you want to look into better enzymes to prepare a library. Okay,
great. Thanks, Damien and Conrad. Good insights there. Two-pronged question here for you, Sarah. What are some of the challenges for hypergenomic sequencing? And two, does traditional Sanger sequencing remain as the go-to option for sequencing?
Interesting questions. So I think with hybridomic sequencing, the main issue you have is trying to find your different populations and variants. So if you're looking for the major population, or if you're really trying to identify what the minor populations are in your libraries and screenings. So depending on your objective going in is going to determine which technique is going to be right for you as a researcher. If you're really just interested in the major population you're looking at, Sanger sequencing is a great option to identify those. However, if you're trying to find those minor variations, you're going to need something that can really identify specifically the quantity of the different populations that are there. And that's where NGS is going to be a great technique for you. So really it comes down to what is your outcome that you're trying to resolve, what is the question that you have, and what is the best technique that's going to answer that. So if you have a major population, you just want to see what that is, go with Sanger. If you really want to hone in on those minor variations, NGS is your best answer.
Okay, good. Thanks for that comparison there. A couple more questions here. I'm just going to open it up. One being, how do you guys see display technologies expediting the antibody discovery process? And as a follow-up of that, how can sequencing technologies be used to enhance traditional phage display selection? Any of you guys want to comment on that?
And maybe I can start on that, because I still remember in the old days when we do the phage display technology, like Sarah mentioned, we only do Sanger sequencing, which means we do a transformation, we pick like thousands of colonies, we individually transformation, make mini preps, and do cycle sequencing on that. It's very labor intensive, time consuming, and very costly. So it costs a lot of money to do the cycle sequencing. I think with the new NGS technology, you can speed up the whole process and with much less money, so you can finish the whole project, knows what exactly in that library was antibody sequences in there. But also I want to add on that Sarah's comments on that. With the NGS technology, you not only see the major species of that library, you see pretty much the whole picture of the library, even though for those rare species in the antibody sequence library, sometimes those rare species may not be the bad hit. They might be very important in the final screening of the antibody.
But I think NGS not only applying to phage display, actually it also can be applied for traditional hybridoma, or like a modified type of like repertoire analysis, right, to develop antibodies. And I think that's also a trend people have been talking a lot. At least what I heard from that is it really more biological and make us more likely to develop therapeutic antibody if we use animals, right? Instead of doing phage display, that pretty much still everything's done in vitro. So I think one way or the other, I think NGS definitely can help out a lot in the antibody development field. So.
Okay, so I have one final question for the panel here. And again, it's open to any of you guys. How has the evolution in DNA reading and writing technologies impacted antibody discovery?
That's an interesting question as well. I think with the advent of the technology that we have, it's really impacted the turnaround time and the cost of going from your antigen to your actual antibody that can help cure disease. So with the advent of NGS and synthetic libraries, being able to create those, you can reduce the amount of time that it takes and the cost it takes to get to that end answer. So it's really helping researchers both in pharmaceutical and industry speed up that process.
To build on to what Sarah said, and also Meow, it's making screening much better. So before customers or scientists would have to do screening and identify targets that may not have been the right ones, but they were limited on the options that they had. So now with, for example, either more screening through Sanger or next generation sequencing technologies, you can actually do much higher throughput, much more sensitive screening of target sequences that you can go back to and test again and again, versus previous methods where you just had to move on through see, what antigens or what sequences were binding to antigens and going from there. So it's much more sensitive. It allows customers to really do a more in-depth screen and hopefully identify the right targets.
Okay, thank you.